Hello, my name is Elizabeth Wood, and I will be talking to you today about an article titled Heterochromatin Formulation and How It Promotes Longevity and Represses Rhomosome RNA Synthesis. First, I would like to present background information. Organismal aging is accompanied by the accumulation of damage to the DNA and other micromolecules and a progressive decline in vitality and tissue function. Some molecules used to explain aging phenomena are free radical theory of aging, heterochromatin loss, telomere shortening, genomic instability, nutritional intake, and the growth signals, and etc. In the chromatin loss model of aging, via Panut proposed that heterochromatin domains are gradually lost with the aging, which results in the silencing of genes and aberrant gene expression patterns associated with old age. So here are some questions that I would like you to think about throughout the presentation. With your current understanding of heterochromatin, do you think it has a direct effect on aging? Second question, do you think the same effects found in Drosophila would be found in humans? There are several studies conducted throughout the article. The experimental tests have produced controversial results such as cellular senescence is associated with an increase in localized heterochromatin formation in the form of senescence-associated heterochromatin foci, hallmark of senescence of aged cells in the cultured and have also been found in aged animal cells. It also shows the premature aging disease in human and animal models correlated with global heterochromatin loss. In this paper, they genetically test the role of heterochromatin in Drosophila aging. They found that the heterochromatin levels indeed affect animal lifespan and that heterochromatin repress, among other things, rRNA transcription. Loss of heterochromatin thus leads to an increase in rRNA transcriptions, a rate-limiting step in chromosome biogenesis and protein synthesis. So the overall hypothesis of this experiment is, do the levels of the heterochromatin prolong the effects of aging? The reason for carrying out this research is because studies have shown that the lack of heterochromatin has promoted aging, so they are seeing if there is a correlation. Now for the role of the heterochromatin. Heterochromatin is important for chromosomal packaging and segregation. It is very important for the genome stability. It has been shown that in Drosophila that heterochromatin is essential for maintaining the stability and repeating the DNA sequence and of the rDNA lotus in particular. Loss of the heterochromatin causes the disruption of the nuclear morphology and formation of the extrachromosomal circulation DNA. The disruption of the nuclear morphology and formation of the ECC DNA has been shown to increase aging in yeast. Positive role of the heterochromatin formation in promoting longevity. The aging animal. To understand heterochromatin's role in an aging animal, the heterochromatin levels in the Drosophila were altered. Heterochromatin protein 1 or HP1 levels and JAK/SAT signals were manipulated. Effects on aging were assessed, and the results suggested that heterochromatin formulation positively contributed to the preventing aging and suppressing illegitimate recombination of the rDNA lotus and unnecessary rRNA synthesis. Now moving on to the experiments of the article. To discover the link between heterochromatin levels and longevity, the lifespan of flies with reduced and increased levels of HP1 was examined. By reducing HP1 levels by half, it caused a dramatic shortening of lifespan compared to the isogenic controls. Moderately overexpressed of HP1 significantly expanded lifespan, resulting in 23% increase in a median lifespan and a 12% in a maximum lifespan. Findings suggest that heterochromatin levels significantly influence lifespan, and moderately higher levels of heterochromatin promoted longevity. This image and graphs support how the increase of HP1 prolongs lifespan. The red line shown is the HP1, so you can see that the flies expressing more HP1 will live longer. Flies heterozygous with the loss of allies were shorter. The second experiment talked about how heterochromatin controls rRNA synthesis. Investigated whether heterochromatin loss also can lead to the depression of rDNA transcription. It measured pre-rRNA transcription levels in flies, found that flies where HP1 is moderately expressed contain more than 50% less pre-rRNA. Differences in body size may not be solely regulated by the rRNA transcription rate. Results are consistent with the idea that changes in the rate of the rRNA transcription may impact global protein synthesis and thus the growth of an organism. The following are methods and experimental techniques used on the Drosophila throughout the article. The first one, which is videotaping, to observe the altered lifespan in flies, they video recorded them, then played video playbacks to show that aged flies exhibited a gradual loss of mobility and eventually became immobile. The second one is chromatin immunoprecipitation, or CHIP. Adult flies of appropriate ages were snapped with frozen liquid nitrogen and then were cross-linked with a 1.8% formaldehyde. Then the flies were regulated in a cell buffer and the CHIP were performed. Then, lastly, the Western blot were used to detect specific proteins, in this case the heterochromatin, in the sample of the tissue of the Drosophila. This research is relevant to us or the public because of the similarities effects of the heterochromatin on muscle function in both the humans and Drosophila. After investigating and monitoring lifespan of the flies with different heterochromatin levels, it was discovered aged flies exhibited gradual loss of mobility and eventually became immobile. Those with reduced heterochromatin levels lost mobility much faster than those with increased levels. Loss of muscle function and frailty in aging humans is similar to the aging Drosophila. So in conclusion, maintenance of the heterochromatin levels is essential for the maintenance of muscle structures and function in which has a direct effect on lifespan. And that concludes my presentation. Thank you.